Hello everyone, so today we're looking at the electron transport chain, or the ETC. So far in glucose oxidation we've covered glycolysis, pyruvate dehydrogenase conversion to acetyl-CoA, and the citric acid cycle. But there's one more step of glucose oxidation, the electron transport chain. The goal here is to describe the transfer of hydrogen ions and electrons in the electron transport and the process of oxidative phosphorylation in ATP synthesis. So here's a big overview picture of the three steps of glucose oxidation. So again, we have our glycolysis, which is going from glucose to pyruvate. And this pyruvate enters into the Krebs cycle or citric acid cycle. And then this produces some ATP as well as a bunch of electron carriers. So here we have our electron carriers, the 3 NADH and 1 FADH per 1 pyruvate. And we also have NADH produced in glycolysis. And then these are all moved into the electron transport chain and oxidative phosphorylation. And through oxidative phosphorylation, we form a lot of ATP. So the electron transport chain is located in the inner mitochondrial membrane. So here we can see the inner mitochondrial membrane. So the overview of the ETC. So essentially the reduced coenzymes NADH and FADH produced from glycolysis and oxidation of pyruvate and the citric acid cycle are oxidized to provide the energy for the synthesis of ATP. In the electron transport chain, aka respiratory chain, the hydrogen ions and electrons from NADH and FADH are passed from one electron acceptor or carrier to the next until they combine with oxygen, which is referred as the final electron acceptor, to form H2O. In the electron transport chain, this couples the electron transfer with hydrogen ion movement across the inner mitochondrial membrane from the matrix. This creates an electrochemical proton gradient that drives the synthesis of ATP through something called ATP synthase, which we'll talk about later. And this is our ATP synthesis. This is our ATP synthase molecule, but we'll talk about that later. So these are those 4 subunits that we discussed. So these are those acceptors or carriers, essentially. And we can see here that we have that gradient being built. So here's that concentration gradient, the high concentration and a low concentration. And we use this gradient to produce ATP. And again, we'll get into more detail later. So here are the results of our steps so far. So from glycolysis, we formed 2 ADP and 2 NADH. We had the oxidation of 2 pyruvates, which gets 2 NADH. The citric acid cycle with 2 acetyl-CoA forms 2 ATP through GTP, 6 NADH, and 2 FADH. So the total ATP yield from these 2 reactions is 4 ATP, 10 NADH, and 2 FADH. So what's happening in the electron transport chain? So essentially we gain these electrons through food. As they're broken down, we get those NADH and FADH. And they move from a high energy state to a low energy state. So they have a large potential energy, and they slowly move down this energy gradient as they're being passed from the different carriers. And as they're being passed, this releases energy for synthesis of ATP. And again, the final thing is oxygen. The electron binds to oxygen, forming water. So here is essentially a close-up of the electron transport chain. So here we can see in the first complex 1, NADH becomes NAD+, so the electrons are going through. So, and NADH will enter at complex 1 where FADH enters at complex 2. And we can see that complex 2, we go through coenzyme Q. Complex 3, we have more hydrogen ions being pumped. So again, we're building that gradient up here, the high concentration gradient. Then through cytochrome C, merges with complex 4. And then we have oxygen and the 2 hydrogen ions forming H2O. So in the electron transport system, there are 4 protein complexes, as I said. 1, 2, 3, 4, bad 3. and 2 mobile carriers. These mobile carriers are cytochrome C, that's the one mobile carrier, and then, coenzyme Q, which is also used like that, and cytochrome C. So these the electron protein complexes are membrane proteins involved in accepting and transferring the electrons carried into the electron transport chain by the reduced molecules such as NADH to oxygen, which is the final electron acceptor. The 4 protein complexes are fixed, so they have names. So complex 1 is NADH dehydrogenase, which accepts electrons directly from NADH. So as I said, NADH goes to complex 1, where FADH will enter at complex 2. So complex 2 is called succinate dehydrogenase, which accepts electrons directly from FADH in complex 1. Complex 3 is cytochrome BC1, which accepts electrons directly from complex 2. And the 4th complex is cytochrome oxidase, which accepts electrons directly from complex 3. So it kind of moves in a chain like that. Complex 2, so 1 to 2 to 3. And again, we had our carriers in here and here. Those special carriers, but we'll talk about those soon. So the mobile carriers in electron transport chain. So there are 2 mobile carriers. There's ubiquinone, also known as coenzyme Q10, which you can see is actually something you could buy as a supplement. So ubiquinone or coenzyme Q receives electrons from complex 1 and complex 2, which carries it to complex 3. Coenzyme Q carries 4 electrons in total from complex 2 to 3. The next mobile carrier is cytochrome C, or we saw it as short form there. It carries 2 electrons from complex 3 to 4. These carriers carry electrons between protein complexes. So again, complexes are arranged in the inner mitochondrial membrane. So again, complex 1 is NAD dehydrogenase because it receives NADH. And we can see NADH enters, and then we get NAD back, which is recycled. And same as here, we get our FAD back, just recycled. Again, so here we have coenzyme Q carrying electrons from 1 and 2. So 1 from NADH and 2 from FADH. But also some are transferred from 1 to 2. And then coenzyme Q carries to complex 3. In complex 3, electrons are carried through cytochrome C to complex 4. And again, complex 4 is cytochrome C oxidase. Complex 3, cytochrome BC1 complex. And complex 2, succinate dehydrogenase. So as we discussed, as this happens, we're slowly building this concentration gradient. So here we have 4 hydrogen ions, 4, 2, and we slowly build that up, and then we build that high to low concentration, and then these will funnel through at high speed due to the strong concentration gradient through ATP synthase, help with the reaction of producing ADP and inorganic phosphate to ATP, and this is where we get that ATP formation. So again, our NADH goes to NADH goes to NAD plus through complex 1, where complex 2, where FADH enters, becomes FAD. Then we have 2 hydrogen ions go through with 1/2 O2, forming that water. So again, the electron transport chain oxidizes NADH and FADH2, involves the passing of electrons, and this allows energy for hydrogen ions to pass, causing that electrochemical gradient that we talked about here. So complex 1, NAD dehydrogenase. So the electron transport chain begins with hydrogen ions and more electrons are transferred from NADH to complex 1. The loss of hydrogen from NADH regenerates NAD plus to oxidize more substrates in oxidative pathways such as citric acid cycle. So as I said, all these products are recycled. Hydrogen ions and electrons are transferred to the mobile electron carrier, coenzyme Q, which is an acceptor, forming coenzyme QH2. Coenzyme QH2 carries electrons from complexes 1 and 2 to complex 3. So for every NADH oxidized, 2 electrons are transferred to coenzyme Q, and this equals 4 electron hydrogen ions pumped through the complex. So here we can see that. So there's a series of proteins and stuff like that within these, such as our iron, but we'll talk about that later. So essentially, the formula is essentially NADH plus a hydrogen ion plus coenzyme Q results in our NAD plus and a coenzyme QH2, which then is, we can see here, QH2, and then that causes the movement of 4 hydrogen ions. So complex 2. This is similar complex and does not span the membrane. So we can see a lot of these are transmembrane, or span the entire membrane, but complex 2 does not. So coenzyme Q obtains hydrogen ions and electrons directly from FADH. It becomes coenzyme QH2 to bring to complex 3. 2 electrons are transferred from the mobile carrier coenzyme Q through the reaction below. The electron is carried via coenzyme Q to complex 3. So again, coenzyme complex 2 does not span the membrane, receives stuff directly from FADH2. This goes directly to coenzyme Q, carries it to complex 3, sorry. So complex 3 and cytochrome C. So the complex 3 complex spans the entire membrane and receives 2 electrons from coenzyme Q. It contains iron molecules, which are reduced to iron ions and then oxidized to Fe3 plus, and which equals 2 hydrogen protons. So this generates energy from electron transfer to pump 2 hydrogen ions from the matrix into the intermembrane space, increasing the hydrogen ion gradient. So remember the gradient that we're talking about, building those hydrogen ions. So essentially, here's the formula: coenzyme QH2 plus 2 cytochrome C is oxidized. So this is oxidized, so then we have iron, Fe3. Then we have coenzyme Q being released, and our 2 hydrogen ions and our 2 cytochromes, and is reduced. So again, 2 transformed electrons causes an electrochemical potential to move 4 hydrogen ions across the membrane. So here we can see 4 hydrogen ions, and here too. So complex 4. Complex 4 spans the entire membrane, and 4 electrons from 4 cytochrome C are passed to other electron carriers, which is oxygen, which is the final acceptor. Electrons combine with hydrogen and ions and oxygen to form 2 molecules of water, so H2O. Energy is used to pump 2 hydrogen ions. 2 hydrogen ions, more hydrogen from the mitochondrial matrix into the intermembrane space, further increasing that hydrogen ion gradient we've discussed. Again, here's a closer look. So we see we need that oxygen to come in and form H2O. And again, this results in more hydrogen ions being pumped out. So again, the complexes are arranged in the intermitochondrial membrane. And here's just an overview again. We have our complex 1, 2, 3, 4, coenzyme Q, and cytochrome C. So essentially, this kind of works on a chemiosmotic mechanism. So essentially, 1, we have reduced substrate, or the fuel donates the electrons. 2, the electron carriers pump hydrogen ions out as electrons flow to O2. 3, the energy of the electron flow stored as electrochemical potential. So they want to move, but they can't yet. And 4, ATP synthase, which is this enzyme, uses the electrochemical potential to synthesize ATP. So it allows these to come in at high speeds and create energy. So the electron transport chain sets up a hydrogen ion gradient, or an electrochemical gradient. This creates a chemical gradient through hydrogen protons, and electro gradient through the positive and negative charges. The electrochemical gradient and electrochemical potential. So the proton pumps are complexes 1, 3, and 4, not 2. So remember, this produced 4, this produced 4, and this produced 2, and this produces 4. Remember, so complex 2 will be here. This doesn't span the membrane, and this does not transport hydrogen ions. Again, we're building that gradient, and then they come through here, and then we have our ATP synthase. So the proton motive force. So this is essentially the kind of mechanism of how the ATP synthase works. So the flow of electrons from NADH and FADH2 causes protons So those are our hydrogen ions. To be pumped from the matrix side of the inner mitochondrial membrane to the outside of the membrane. The movement of protons is accomplished by the complexes of 1, 3, and 4. The protons then move back into the matrix through the FO, F1 ATP synthase. So a lot of these pieces will named F something, F with a number, and we'll talk about that later. But so these, again, they'll move back into the matrix through the ATP synthase, driving ATP synthesis. So again, the electrons donated from NADH and FDH are moved through the complexes, and this causes the movement of protons in complex 1, 3, and 4. And again, they move back in, essentially making the ATP synthase work. So essentially, it is ATP and inorganic phosphate plus the energy from the movement of those hydrogen ions and through the molecule ATP synthase or through the complex forming ATP. So this process of oxidative phosphorylation couples the energy from the electron transport chain to the synthesis of ATP through ATP synthase. So as we mentioned, there's different units. So we have our F0 unit, which conducts protons, which causes F1 to bring ATP into contact with inorganic phosphate to form ATP. So F1 is essentially like kind of the head, and F0 is kind of the transmembrane part. So there are approximately 3 protons are needed to synthesize each ATP. So the movement of protons across the membrane. So as we mentioned, hydrogen ions re-enter the matrix through ATP synthase. The influx of hydrogen ions is coupled to ATP synthesis. So again, we keep building that proton gradient. We have a membrane potential. Again, here's our F0 unit attached to the membrane. Our F1 unit is kind of the head. We need our ATP and ADP and inorganic phosphate to form ATP. Again, the hydrogen ions are moved through, essentially to create that energy for this reaction to occur. Again, these protons, after they go through ATP synthase, the protons are ejected by electron flow to respiratory complexes, which is right here. The transfer to ADP into the mitochondria and ATP out. So ATP is synthesized, has to get out of mitochondria into cytoplasm. And ADP and inorganic phosphate has to get into the mitochondria from the cytoplasm. So this is through antiport and symport mechanisms. So we have our ADP-ATP translocase, where they kind of swap out, where an ATP goes in and ATP goes out. So they kind of work in an antiport mechanism. So they use each other's gradients to move in and out. We also have phosphate translocase, which is a symport, where inorganic phosphate and hydrogen ion can move in together. So this occurs in the complexes. So when we move those hydrogen ions in, inorganic phosphate can come with it. And then again, we use these respiratory complexes to remove hydrogen ions once they go through ATP synthase. So again, funneling the gradient. So ATP production uses 4 protons. ATP synthesis requires 3 protons through ATP synthase for 1 ATP. The transport of ATP out, it involves the transport of ATP out and ADP into the mitochondria, causing 1 proton equivalent. So that was through our symport mechanism, which we're talking, or antiport, sorry. Again, there are 4 net protons for cytoplasmic ATP. So even though it's 3 protons through ATP, it ends up more likely being 4 net protons for 1 cytoplasmic ATP. Essentially, because you have to use 1 to help get it out. As that's why this is highlighted, cytoplasmic. So ATP production from NADH. So as we mentioned, NADH and FADH can both enter. So remember that NADH transfers electrons to complex 1. In total, 10 protons are moved out of the matrix. So 4 through complex 1, 4 through complex 3, and 2 through complex 4, and none through complex 2, as we mentioned. So since 4 protons are needed for 1 ATP, and we see we get 8 or 10, so you essentially do 10 divided by 4, is that we get 2.5 ATP generated per NADH. So for FADH, so know that FADH transfers electrons to complex 2. So in total, 6 protons are moved out of the matrix, 4 for complex 3 and 2 for complex 4. So since FADH goes to complex 2, it does not get the 4 hydrogen ions that are moved in complex 1. So this doesn't happen. So now that's why we get 6 instead of 10. So again, 6 divided by, what was it? 6 divided by 4, I'm sorry. We get that 1.5. So 4 protons needed for 1 ATP. So this means we need 1.5 ATP. We have 1.5 ATP generated per FADH. So the regulation of the ETC and ATP synthase. So the regulation of this is based on the availability of ATP and inorganic phosphate, oxygen, and NADH. So one simple example is that if we don't have oxygen, this won't occur because we need that final electron acceptor. Anyway, so the electron transfer chain ATP synthase decreases with low levels of any of these compounds and decreases with the formation of ATP. So if we have tons of ATP, it's not going to keep wasting energy to produce it when we already have it. So when a cell is active and ATP is consumed rapidly, the elevated levels of ADP, because it's being broken down, so ATP going into ADP and inorganic phosphate, this will activate the synthesis of ATP. The activity of the electron transport is strongly dependent on the availability of ADP for ATP synthesis. So NADH, so as we talked about, NADH enters the electron transfer chain via the glycerol phosphate shuttle. So remember that NADH made in glycolysis has to get into the mitochondria in order to reach the electron transfer chain. This occurs via the glycerol phosphate shuttle. The glycerol phosphate shuttle transfers the electrons from the cytoplasmic NADH to mitochondrial FADH, which yields 1.5 ATP. This means that if the glycerol phosphate shuttle is used, the cytoplasmic NADH will yield 1.5 ATP because the FADH enters the citric acid cycle complex 2. In comparison, the yield of ATP from oxidation of a molecule of glucose to CO2 and H2O. So, starting from glycolysis, we have 2 ATP are used, 4 are gained, so there's a net of 2. We have 2 NADH, 3 or 5, depending on the shuttle. So we have our glycerol phosphate or malate aspartate, which isn't here. I guess the slide got deleted. Sorry about that. So this would be our glycerol phosphate because we have 1.5 times 2 in comparison to the other one, which would be 2.5, which is malate aspartate. Then we have our pyruvate dehydrogenase reaction where we have 2 NADH per glucose, which equals 5 ATP. Then we have to the citric acid cycle, we have 6 NADH, which yields 15 ATP. Then we have succinate to fumarate, which gives us 2 GGP, which is equivalent to ATP. Then we have our 2 FDH, which gives us 12 protons, so 12 divided by 4 would be 3 ATP. So our total, depending on the pathway here, can be either 30 or 32 ATP. So summary. So here's a summary of the entire process. So here we have a glycolysis to pyruvate. Pyruvate enters. Again, this is giving us an example where we can also see fatty acids enter in the different stages. But again, we have a citric acid cycle again, acetyl-CoA formation, and then there's electron transport chain in our ATP synthase. So summary, they're 4-phase process, and there are 4 complexes. There's a total yield of 30 to 32 ATP. And this is for 1 glucose. Demand for ATP drives predominant fate of pyruvate, which remember we had a pyruvate with a crossroad, which is a important step in our metabolism. And this process is regulated by NADH and NAD and ADP to ATP ratio and O2 and inorganic phosphate. So, I think this slide got mixed up. But again, as I mentioned, there's the malate aspartate and also the glycerol phosphate. So again, this was the other pathway, and the NADH made in glycolysis can also enter through the malate aspartate shuttle. The MA shuttle transfers electrons from cytoplasmic NADH to mitochondrial NADH, which yields 2.5 ATP. This means that if the malate aspartate shuttle is used, the cytoplasmic NADH will yield 2.5 ATP because the NADH enters at complex 1. So I'll just go back. So again, this would be the malate aspartate. Whoops, malate aspartate. And that would be the glycerol phosphate. Again, this would enter at complex 2. This would enter at complex 3. Again, I'll end on the summary slide here. And then again, I recommend going through some of these, like, I really recommend focusing on the proton movements. And so some of the stuff like this, because that's really important. And definitely get the complexes down. And then, yeah, so this is, say, the big slide. Because a lot of stuff will test you on just what you're actually yielding. So sometimes a lot of places won't test you on like the specific enzymes or what's happening within each complex. They're just going to want you to understand the actual process, and understanding what's gained and what's being used is really important. And I think this is one of the better slides to help you. So we have our final couple lectures coming up on fat metabolism, and I'll see you in that one. I hope you guys enjoyed it. Take care.